Yeah, you've already heard my title, so we'll start right away to save some time. So first, I would like to say a few words about what is a tardigrade. Tardigrades are those small invertebrates. It's a pretty large group of animals. Here you can see there is a mouth, there is a back end of the body. They're not very big, usually not bigger than 1 millimeter. That would be very huge for them. However, they are most relevant for our topic here today because of their ability to withstand all kinds of hazardous conditions at every stage of their life cycle. So with that introduced, let's talk about the other part of my work. So what is cyanide and what does it do? In very short answer, cyanide kills you. It kills you very badly and very deadly. But to be a bit more precise, how does it work? Here you can see a schematic of respiratory chain that is present in mitochondria of most cells, at least of nucleic cells. And here you can see that cyanide affects the fourth complex of this respiratory chain, preventing proper functioning of this whole molecular machinery. Essentially, it stops functioning of mitochondria, which usually leads to death of the organism. It simply cuts off its power supply, we could say in more analogous terms. Yes, and it's cool, but why should you care about it from perspective of astrobiology? That is because tardigrades, when they are put into cyanide, they stop. They seem like they are dead. But very quickly, even after 15 minutes, after you, of course, take them out from the cyanide and put them back into water, they start moving again. They basically look like they are perfectly fine. And again, this is cool, but why is it relevant for astrobiology? As some of you, probably everybody, has heard the joke that the mitochondria is the powerhouse of the cell. And that is true to an extent, in that mitochondria are extremely crucial for functioning of any organism. And when you are thinking about astrobiology in terms, for example, of long-term preservation of living tissue, for example, for a long-range travel that would take a lot of time, you will be trying to find and eliminate points of failure. And mitochondria, because of their crucial nature to the functioning of organism, are one such point of failure. And I believe that eliminating this weakness, you could say, is very important if we want to even dream about preserving life across a long time and also very long distance travels. So what exactly happens to confer tardigrades this quite staggering resistance? Because the concentrations that we are talking about here it's hard to, of course, compare humans to such small animals, but they are relatively hundreds of times more powerful than the dose that could be deadly to a man. And of course, for humans, we just need to eat this substance, and they are pretty much dipped inside the solution. So tardigrades, we suppose, possess alternative oxidase. This is this component in green as a part of the respiratory chain, which, to shorten its to its digestible explanation, serves as a bypass. Essentially, when this fourth complex becomes blocked by cyanide, this AOX complex is taking up its role, and therefore the cell can function normally, or at least somewhat normally, in comparison to cells that lack it and would be simply dead. So how did I test this resistance? First, I took tardigrades and added to them cyanide solution, potassium cyanide solution. This resulted in their apparent death. They stopped moving, they became like very stretched, looked exactly like dead animals. And then they were put back into clean water and they were observed in time. And in pretty much every case, they came back alive and kicking. So what I was doing for the most part was recording this time of recovery. You can see it on the y-axis. It's in seconds. And I compared mostly two species. One is carnivorous and more dry habitat inhabiting species, while the other one is more aquatic species and also tends to eat algae. Both of them performed exceptionally well. As you can see, their time of recovery was extremely fast. For this second species, it was a bit faster than for the other one, but it's still exceptional to see it in any animal. However, it's cool when we observe them for 15 minutes, 20 minutes, even a few hours. But important question is, does it have any cost? Does it incur some lasting effects that can then reflect on this animal, maybe even in few generations? So in order to test that, at least to some extent, I tried exposing tardigrades from the same species to different doses of potassium cyanide, and then keeping them in cultures, like single specimen in each dish, to see if they will start dying off, if their survival will be affected by and fitness will be affected by this incident. So to stress it once again, because it's very easy to mix it, in this case, they were subjected only for one time, for like an hour, to the poison, and then they were put to water. And as you can see, at first it was all good. There were no changes. However, after around a week, especially the highest concentration began to die off. And while control group and the lower concentration didn't seem much different from each other, you can see that with higher concentration, the animals started to die off faster and faster, and their fitness was, as a result, much worse. So this would suggest that there is some cost, that this ability is not as foolproof as it would seem at the first glance. So in order to check what is happening, we decided to slice them open and see what's inside of them. And here you can see photos from a transmission electron microscope from control group and from this specimen that was exposed to high concentration of cyanide. And this is photos taken 24 hours after a 10-minute exposure. And what you can see with naked eye are those huge autophagic structures. So here is unfortunately not very well visible. Here those arrows indicate mitochondria. But this poisoned animal, even though it was outwardly perfectly fine, there was some serious issues going on with its cells. And it's very likely that those may be contributing to later death of those animals in this long-term effects test. So yeah, it's fine and good, but it's still very open research. What I would like to do next is first to see some more long-term effects. For example, right now we are waiting for results with also transmission electron microscopy slices of animals 30 days after exposure. So it's a much longer time span, time span. So I'm hoping to see if that damage that was incurred by poisoning is either repaired, or maybe it actually goes further and the condition of the animal worsens. And also, as you can see, cyanide blocks complex 4 of the respiratory chain. And as the name suggests, there's a lot of more. And what I would like to do is to test cyanide in combination with inhibitors of other complexes to see how those animals will react to it. Especially, of course, of crucial interest for me is this AOX, because as I told you, it creates a bypass for this respiratory chain. So it would be like plugging a hole. We'll see if adding inhibitor of this complex as well will kill those animals finally, or maybe they will still survive, and that would be also very interesting situation. So thank you for listening to my presentation, and I'm very glad to be part of this conference. Thank you.